Warm greetings to all 4th semester botany students. I'm Dr. K.Y. Pratibha. In this session, I would like to talk on anther wall layers. In the previous session, we had studied about the development of the anther. In angiosperms, anther is a bilobed structure containing 4 microsporangia, so it is known as tetrasporangiate. The anther has following layers. The outermost layer is known as epidermis. Inner to the epidermis is the layer of endothelium. Inner to the endothelium there are 2 to 3 layered middle layer structure that usually disintegrate during anther maturation. The innermost layer is known as tapetum. Inner to the tapetum are microspore mother cells which give rise to the pollen grains through meiotic divisions. So if you see this picture, you will understand the development of the anther. In the beginning, anther primordia will emerge from the stamen, and that anther primordia will have a homogeneous mass of the cells. In that mass of cells, 3 layers differentiate, L1, L2 and L3. The L1 differentiates into epidermis, and then some part of the epidermis becomes stomium during the dehiscence of the mature anther. L2 becomes, in the L2 region, archisporial cells differentiates and will form primary parietal cells which will form the anther wall, and primary sporogenous cells which will form the microspore mother cells, and eventually which will form the microspores. The L3 will form the connective and vascular bundle. So this is the schematic diagram of the development of the anther and its wall layers. If you see this picture, you will understand the types of anther layers that are present in the anther. The outermost layer is epidermis, followed by endothelium, followed by middle layer, and the tapetum is the innermost wall layer of the anther, which will encircle the microspore mother cells. Now let us study anther walls one by one in detail. Epidermis is the outermost single-layered protective sheath of the anther. It divides anticlinally and tries to keep space with the enlarging internal tissues of the anther. It tries to keep pace with the enlarging internal tissue of the anther. As a consequence, the epidermal cells undergo considerable stretching and flatten in surface area. It provides structural integrity of the anther, assists in gaseous diffusion, prevents moisture loss, and in the dehiscence of the anther lobes. Sometimes the epidermal cells may be cutinized or lignified. So the first wall layer of the anther is epidermis. It is protective in its function. It is single layer, and in young anther it will be very broad, but in mature anther it becomes flattened. The second anther wall layer is endothelium, formed by the primary parietal layer which divides into secondary parietal layer, which again will form the endothelium, undergo division and form the endothelium. Endothelium is the outermost layer of the descendants of the parietal cells located immediately below the epidermis and are called as the endothelium. So epidermis will be there from the beginning of the anther development, and endothelium is the product of the primary parietal layer. It attains the maximum development before the dehiscence of the anther. Endothelium cells are radially elongated and decorated with fibrous bands. Hence, this layer is called fibrous layer and are hygroscopic in nature. And these fibrous bands run upward from the inner tangential walls, ending near the outer wall of each cell as an incomplete ring. The outer tangential walls remain thin. The endothelium is associated with high proportion of alpha cellulose and small amount of lignin at maturity. The specialized nature of the endothelium together with the stomium helps in the dehiscence of the anther. So the second wall layer is endothelium. It is characterized by the presence of fibrous bands which runs from the inner wall, tangential wall, to the outer region of the cell, and these fibrous bands are cellulose and lignin to some extent, and they help in dehiscence of the anther along with the stomium. The third wall layer is middle layer. Below the endothelium, 2 to 3 layers of the cells are present which constitute middle layers. Usually one middle layer is present as in dicots and monocots, but sometimes it may be 2 or 3, or complete absence of middle layer can also be seen. The cells of the middle layer lack the capacity to divide and hence cannot adjust with expanding sporogenous cells. As a result, it gets crushed and cannot be seen in mature anther. The cells of the middle layer are usually epimeral. The layer adjacent to the endothelium may even develop fibrous thickenings. The middle layer, which is adjacent to the endothelium, even can develop fibrous thickenings. A few instances, the middle layer acts as a storage center for starch and other reserves that is later mobilized for developing pollen. So the third wall layer of the anther is middle layer. It may be 1 or 2 or 3, depends upon the type of angiosperm plant. Usually one middle layer is present in monocots and dicots. The middle layer cannot undergo anticlinal division afterwards. Once it is formed, it cannot further undergo division. So when the anther, the sporogenous tissue is expanding, the middle layers cannot expand along with the sporogenous tissue. Hence it gets crushed in the mature anther. The function of the middle layer is storage of food in the form of starch and other reserves that is later mobilized or given to the developing pollen. The fourth important anther wall layer is tapetum. So the fourth wall layer of the anther, which is physiologically very important, is tapetum. Tapetum is the innermost wall layer of the anther and usually derived from the inner secondary parietal layer. This we have done in the previous session. So its development, it originates from the parietal layer, and sometimes tapetum may be dimorphic. Some of the cells of the tapetum are formed from the parietal layer, and some of it can be formed from the connective tissue or sometimes from the sporogenous tissue also. The tapetum surrounds the sporogenous tissue and attains maximum development when the microspores are in tetrad stage, after which they go into decline, and that results in the collapse of the cells. Usually tapetum consists of a single layer, very common. Very rarely it can be having 2 to 3 layers. The tapetum completely surrounds the sporogenous tissue. Major part of it is derived from parietal cells, and a small part can be developed from sporogenous tissue or from the connective tissue. Tapetum transports the nutrients to the developing sporophytes. So physiologically tapetum is very important. Physiologically, because it is very important because it transports food materials to the developing microspore mother cells, which undergo meiosis to form pollen grains, and for the developing pollen grains also the nutrition is supplied by the tapetum. Characteristic of tapetal cells, they are distinctly enlarged or radially elongated and always epimeral. So in a completely mature anther, we will never see the tapetal cells because it would have given its food to the developing microspores, and hence after the anther is matured, it disintegrates. The cytoplasm is dense with prominent in the young tapetum or the young anther. The cytoplasm of the tapetal cell is dense with prominent nuclei. It contains ribosomes, mitochondria, endoplasmic reticulum, many vesicles and other active organelles like Golgi complex, dictyosomes, like that. Cells may be multinucleate or polyploid and are comparatively rich in DNA. Tapetal nucleus behave very abnormally. It can be multinucleate and they are comparatively rich in DNA. There is a irregular mitotic divisions and nuclear fusion we will see in the tapetal nucleus. They are characterized by rapid and intense activity with the degeneration of their cytoplasm. Now let us go to the behavior of the nucleus in the tapetal cells. So now as far as now we have learned about the fourth anther wall layer is tapetum. It is physiologically very important. It is single layer. It completely encircles the sporogenous tissue. It has a dense cytoplasm and a prominent nuclei, and it is rich in all the cell organelles. And the behavior of the nucleus in tapetal cell is very unique. Tapetal cells undergo dynamic fluxes during their short lifespan. The characteristic cytological feature of the tapetal cells is the increase in the content of their DNA. Since DNA increase is not followed by regular mitotic division, it results in cytological abnormalities like multinucleate cells. So, sometimes many nucleus we will see in the tapetal cell, or endomitosis, where the chromosome go on dividing without the cytokinesis. So many chromosomes we will see in the tapetal cell. Polyteny, that is many strands. A chromosome is supposed to have 2 strands, but many strands can be seen in the chromosomes. So chromonemata increases, that is called polyteny. Formation of restitution nucleus is also reported in tapetal cells. So each cell will undergo cell division, but at anaphase the cell division stops. So that can also be seen in the tapetal cells. So tapetal cells, if you see this picture, you will understand that the tapetal cells shows the tapetal cells and its nucleus behave in an abnormal way. Its nucleus can have multinucleate condition, endomitosis or polyploid chromosomes, polyteny, and formation of restitution nucleus are all common in tapetal cells, or the nucleus of the tapetum. Now let us go to another important aspect is the types of the tapetums. Types of tapetum on the basis of behavior, there are 2 kinds of tapetum. So on the basis of behavior, at what time the tapetal mass breaks down, depending on that, there are 2 types of tapetum: amoeboid tapetum, or the other one is secretory or parietal or glandular tapetum. Amoeboid tapetum and glandular tapetum. Amoeboid is also called as invasive or periplasmodial tapetum. Here, the tapetum is characterized by an early breakdown of the inner and radial wall of its cells. The protoplast masses moves into anther cavity, followed by the fusion of protoplast to form periplasmodium, which encircles pollen mother cell before meiosis. Example: Alisma, Arum, Tradescantia, Typha, Helianthus. So, amoeboid tapetum is characterized by the breakdown of the tapetum. The tapetal cells break down very early and it will encircle the developing pollen mother cells. Before the pollen mother cells undergo meiosis, before only the tapetal cells break down and encircles the pollen mother cells. So they form an amoeboid like invasive, or the pseudopodia like amoeba. That is why it is called as amoeboid or invasive or periplasmodial tapetum, seen in Tradescantia, Typha, etc. Secretory or parietal or glandular tapetum, which is the most common type of tapetum. Here the tapetal cells remain in their original position throughout the microspore development, and it breaks down at the time of pollen maturity, that is after the meiosis of pollen mother cells. They secrete their substances from their inner faces. This type of tapetum is most common among angiosperms, around 87%. Example: Mirabilis, Heloborus and Nigella and Nigella. Okay, so these are the examples for the secretory tapetum. So basically we have to understand both the tapetum, whether it is the amoeboid tapetum or glandular tapetum, both will break down. But at what exact time it breaks down, depending on that, we have classified it into amoeboid, which breaks down early, before the division of the pollen mother cell only. If the tapetal cells break down to nourish the pollen mother cells, then it is called amoeboid. Whereas the glandular tapetum, it is going to break, but at a later stage, when the pollen mother cells have undergone meiosis, when the pollen grains are released, at that time the tapetal cells break down. Till then, they will be nourishing the pollen mother cells and the pollen grains through their inner, inner cells. They will they secrete their substances from their inner faces. Okay. Now let me go to the amoeboid tapetum. So amoeboid tapetum, this type of tapetum is characterized by early breakdown of the walls of the tapetal cells. I had told you this. The inner and radial walls of the tapetum break down by hydrolytic enzymes, and their protoplast containing food materials penetrates between the pollen mother cells and moves into the inner anther cavity. So, very early stage, the amoeboid tapetum breaks down, and it protoplast along with food material, it will move in between the pollen mother cells of the anther cavity. After intrusion, they fuse with each other and forms a mass of tapetal periplasmodium, or shows amoeboid movement around the pollen mother cells and forms the periplasmodium mass. A detailed study has been done in Tradescantia. Amoeboid tapetum is studied in detail in Tradescantia, which shows the reorganization of cell organelles. During meiosis, the periplasmodium releases callase, that is meiosis of the pollen mother cell. These tapetal cells release the enzyme callase, which will degrade the callose wall of the pollen tetrads. This tapetal plasmodium remains associated with the pollen grains till their maturity by providing the nourishment, and when the anther gets drying up, the tapetal periplasmodium gets dehydrated and coated over the surface of pollen grains, thereby helping in the formation of exine. Amoeboid tapetum is considered as the primitive tapetum. Let's go to the second type of the tapetum, that is glandular or secretory type of tapetum. The cells of the glandular tapetum remains intact throughout the microspore development. They secrete their substances from their inner faces. Secretory tapetal cells are thin and possess almost all cell organelles like mitochondria, plastids, dictyosomes, etc. Some spherical structures called proeubish bodies are also present. So glandular tapetum is the most common type in angiosperms. It is rich in all the cell organelles, and very unique characteristic of the glandular tapetum is the production of proeubish bodies, and these proeubish then will form eubish bodies, which will then form the exine wall of the pollen grain. Just before the pollen mother cells undergo meiosis, the walls of the tapetal cells become thick, and there is considerable increase in the number of ribosomes and proeubish bodies with the completion of pollen development. So simultaneously, the tapetal cells will be behaving in its the tapetal cells behavior varies in accordance with the behavior of the pollen mother cells and the formation of the pollen grain. So the way the sporogenous cell is behaving, in accordance with that, tapetal cells also change its behavior. So when the pollen mother cells are ready to undergo meiosis, the proeubish bodies are seen in maximum number. Proeubish bodies pass through tapetal cell membrane and into the space between cell wall and cell membrane, and these proeubish bodies will be coated with sporopollenin, and now they are called as eubish bodies. Eubish bodies are involved in external thickenings of exine over the pollen grains, example higher monocots and many dicots. At the time of pollen tetrad stage, glandular tapetum breaks and encloses the developing pollen grains in the locule. So, glandular tapetum is the most common type, glandular or secretory tapetum. It is seen in 87% of the angiosperms. Here the tapetum breaks only after the pollen mother cell has undergone meiosis, but till then it will be nourishing the pollen mother cells through its secretions through its inner phases. And here we will come across a very unique structure called proeubish bodies, which will be coated with the sporopollenin and becomes eubish bodies, which eventually will form the exine ornamentation of the pollen grains. So let me go to the next very important aspect, that is functions of tapetum. So tapetum is the most important anther wall layer because it supplies the nourishment to the developing pollen grains, and that if the tapetum is non-functional, then that anther becomes sterile. So functions of tapetum: the nutrients are transported through tapetum to the sporogenous tissue, secretion of polysaccharides into the locules during the free microspore stage, which are absorbed by the microspores. So without tapetum, there is no nutrition for the developing microspores. Tapetum is involved in the synthesis of calase enzyme, which release microspores in a tetrad by degrading callose wall. So when the pollen grains are formed, the tetrads will be encircled in a callose wall. So the tapetum will synthesize a calase enzyme which will degrade the callose wall of the pollen tetrad and hence release all the pollen grains. The tapetal cell secretes sporopollenin, which help in the formation of the exine of the pollen wall. So exine is the most protective layer of the pollen grain. So that is formed to 50% of the exine wall, that is sporopollenin, will be contributed by the tapetum. Male fertile and sterile condition is linked to the formation of improper development of tapetum. Cytoplasmic male sterile plants show that faulty tapetum is responsible for male sterility. So without proper tapetum, the anther is unable to produce the pollen grains. So tapetum is very much important for the healthy production of healthy pollen grains. Now, another function of the tapetum: formation of pollen kit and tryphin, which are deposited on pollen surface, which help to bind pollen grains together and for efficient insect pollination. So pollen kit is the contribution of tapetum. This pollen kit is formed by the tapetum, which attracts insects and protect the pollen from ultraviolet rays. Pollen wall containing the pollen grains will have pollen wall, and these pollen wall will have proteins derived from tapetal cells. These proteins are present in the cavities of exine, which recognizes the compatible pistil. So inside the exine there will be proteins which are given by the tapetal cells, which recognizes the compatible pistil during pollen germination. Tapetal cells have other activities that result in the formation of different structures. In Onagraceae, tapetal cells play a very important role in the formation of flexible threads known as Visin threads. In Asteraceae, the tapetum forms acetylysis resistant membrane outside the sporogenous tissue. So let us just revise some of the functions of the tapetum. Number one, the tapetum, the nutrition to the developing sporogenous cells is given by the tapetum. The tapetum will produce calase enzyme which breaks the callose wall of the pollen tetrad. The tapetum helps in the formation of the exine of the pollen wall. Male fertile sterile condition depends upon the activity of the tapetum, and the pollen kit is the contribution of the tapetum, and the pollen wall will have proteins which will recognize the compatible pistil, which is also the contribution of tapetum. In some of the Onagraceae, the tapetal will form threads, or in case of Asteraceae, resistant membrane is produced by the tapetum. Now, let us just revise or compare the TS of the young anther with the TS of the mature anther. So in the beginning slide we had studied the structure of the young anther. So now let us take up the TS, the transverse section of the mature anther. So how the change happens from the young anther to the mature anther is very important. So the TS of the mature anther internally contains 4 microsporangia. So we can see 4 the microsporangia inside the anther. Each microsporangium consists of an outer anther wall and an inner anther locule. The anther wall is composed of epidermis, as all of us know that. But here some changes will happen in the epidermal cells when compared to the young anther, mature anther. The epidermal cells of the mature anther become stretched and flattened in the mature anther. So the epidermis becomes stretched and flattened in the mature anther. The endothecium, these are hygroscopic, and when they lose water, they contract, as a result of which anther dehisces, and endothecium helps in the release of pollen grains. So endothecium cells are very important. They have the fibrous bands which are hygroscopic. When they lose water, they contract and they release the pollen grain. So in the mature anther, endothecium helps in the breakage of the anther. Along the line of dehiscence of anther lobe, the endothecium is composed of thin walled cells called stomium, which helps in anther dehiscence. The third layer, as we know, is mid layer. In a mature anther, the cells of the mid layer becomes flattened and collapse due to the pressure exerted by anther locule. Only in young anther you will see mid layers. Mid layers will be totally absent, or it becomes epimeral, and it degenerates in the mature anther because it cannot undergo the anticlinal division to keep in pace with the expanding sporogenous cells. That is why middle layer will become it becomes disintegrated in the mature anther. Now the tapetum in mature anther, tapetum would be used up for the development of pollen grains. In young anther, tapetum will be very prominent with the with a prominent nucleus and a dense cytoplasm. In mature anther, the tapetum, whether it is glandular or amoeboid tapetum, the tapetum would be broken up. It breaks out to nourish the pollen mother cells or the pollen grains. So that is the reason in mature anther we cannot see the tapetum. So coming to the anther locule, it is the sporangial cavity. Anther locule is the sporangial cavity containing a number of microspore tetrads. So in mature anther, we will see the microspore tetrads or pollen grains, whereas in young anther, we will see sporogenous tissue, intact sporogenous tissue, whereas in mature anther, we will see the pollen tetrads or pollen grains formed from the microspore mother cells by meiosis. So meiosis would have been completed and all the pollen grains will be there in the anther locule in mature anther. So microspores, after the separation from tetrad or pollen grains, and the anther will break open at the region of the stomium to release the pollen grains. Now let's take up the anther dehiscence. The stomium is the region of the anther where the dehiscence occurs. The endothelial cells along the junction of the two sporangia of an anther lobe lack cellulose thickenings. This region is called stomium. The region along with the hygroscopic nature of the endothecium helps in the dehiscence of the anther at maturity. So endothecium along with the stomium helps in the breakage of anther. The cells lose water and uneven thickness cause the cells to stretch to a greater extent. This creates a tension that eventually leads to the anther being split. So let us revise what we have studied in this session. The well-differentiated anther wall comprises an epidermis, an endothecium, 1 to 3 layered middle layer, 1 to 3 mid layers, and tapetum. Epidermis is protective, provides structural support, prevent anther water loss, and specialized epidermal cells in the stomium region split at maturity to facilitate anther dehiscence and release of pollen. Second layer is endothecium, which develops fibrous bands of lignocellulosic secondary thickenings that provide the mechanical force for anther dehiscence. Middle layers are short-lived and get crushed during pollen development, and tapetum plays a crucial role in the pollen development. There are two basic types of tapetum, amoeboid and glandular. Ubish bodies are characteristic feature of secondary tapetum. Tapetum is involved in the supply of nutrition to the pollen mother cells. These are the references. Thank you.